So, last up is also a group leader from DTU, Center for Biosustainability, Morten over here, and he's going to talk about the reverse genetics 2.0. So,
thanks a lot, Niklas. Thanks a lot for inviting me. Fantastic to see so many people being interested in CRISPR-Cas9. So, my name is Morten. As Niklas said, I'm a group leader at Novo Nordisk Foundation Center for Biosustainability. We are basically the spaceship that landed over there 2 months ago, and a lot of water came out of the building. We are also a pretty new DTU institute, formed 5 years ago by a generous grant from the Novo Nordisk Foundation. So I'm going to give you a few examples about basically what we use CRISPR for at the center, but only a few. And mainly in prokaryotes, we've heard a lot about the use of CRISPR in eukaryotic systems. So I'm going to give you, I think, 2 examples of the sort of special case of using CRISPR in prokaryotes. But I also thought that I would take this opportunity and actually give you sort of a, let's say, broader perspective on why I believe that CRISPR-Cas9 is such a groundbreaking technology. So I wanted to start by the word gene editing that we've heard a lot. So basically, that's kind of the most used metaphor for what CRISPR-Cas9 is. So when I say metaphor, what is a metaphor? It's something we very often sort of use to try and describe with a common word, something which is fairly complex in science. So let me expand that metaphor a little bit. So we have, let's see if we can point. Does it work? Doesn't matter. So we have CRISPR-Cas9 hiding down here, and this bigger sort of metaphor. So editing, I actually looked it up today, I didn't really know. I mean, editing actually really originates basically from editing books. It doesn't have, to my knowledge at least, sort of a meaning before the discovery and the development of books. So you can expand that metaphor so that all the other sort of important features of books can be explained as a metaphor for biological systems. So in books, in order to actually write a book, you need an alphabet. So in biology, we have the nucleotide, we have the 4 nucleotides in DNA. You need to decide on a specific language that you have in common with other people. That happens to be more or less universal in living systems. So we have the genetic code, which is pretty much universal for all living organisms on this planet at least. Now you also need to learn to read, and we've heard a lot already about next generation sequencing and something called Sanger sequencing, and I'll go a little bit more details with these inventions later on to sort of put, again, CRISPR-Cas9 in perspective. Now when you can read DNA, it's also important that you can actually write. So in the book industry, you can write by hand, but it's also been extremely important to actually print books. And so a sort of a parallel development has been done in biology, where DNA synthesis has developed enormously over the last 20 or 30 years. Now it's really nice if you have a book and you make a mistake that you can edit your text, right? And this is where CRISPR-Cas9 and other technologies that we've heard about today comes into the picture. So really, what most people talk about when they talk about CRISPR-Cas9 is to correct small mistakes in the language of DNA or biology, right? So, doing corrections to already written text, basically. So that's what we mostly use CRISPR-Cas9 for. I also wanted to highlight something, that is that probably all of you in the audience are capable of doing all these, except for maybe the last part of the book. So we can all, we know the alphabet, we have a language or several languages, we can read, we can write, we can even edit using Word on our computers. But probably not a lot of you are really good authors, right? And it's actually the same thing in biotechnology. So, pretty much this technology has been developed for us in the past 50 years. And if you're biotech students or biology students, you will learn and master some of these technologies, including CRISPR. Doesn't mean that we actually really know how to create fantastic things in biotechnology. Okay, if I knew how to create a fantastic thing in biotechnology, I wouldn't be standing here. I would be on my yacht somewhere, cruising in the Pacific Ocean. But I'm pretty good at these tools. I just haven't really cracked the code and invented something really fantastic in biotechnology. So metaphors are not always perfect. So for example, editing books, I kind of have the feeling that editing DNA is kind of more important. And a really good example is, of course, people with genetic diseases. That's an already written book using this metaphor. If we can go in and correct something that is dysfunctional in a human being, I think that would be an enormously important use of CRISPR-Cas9, where we go in and edit a code that has already been written. I don't think it has had the same impact for books. On the other hand, it might actually be a really good metaphor. There are some scientists in the field who believe that CRISPR-Cas9 is not going to be very important in 10 or 20 years. And the reason why is that the DNA synthesis industry is really taking off in a rapid pace. So the argument is that in 10, 20 years, DNA synthesis is going to be so efficient and so cheap that we don't want to correct DNA. We just write a new code. We don't want to go in and correct DNA in living organisms. We just provide them with a new genome. It seems like a dream right now, but as I will show you some data on, there are actually some indications that this might be true. Okay, so one thing which is kind of a common theme for groundbreaking technologies is that they actually take off in exponential pace. So let's keep in the book industry and note that we have centuries down here on the x-axis, and note that it's a log scale on the y-axis, right? So the increase in the number of manuscripts in Europe over the past many centuries is sort of a straight line in a log plot. So that means it has been exponentially amplifying the number of books in Europe in the past many centuries. Now, something happened in the 15th century. We sort of get a break in this curve. We get another straight line. So still exponential growth, but even faster, and that's because we start printing books and not just writing them by hand. So this is really one of my favorite slides. So some people who know me in the audience know I like this a lot. So this is a comparison of some of the technologies that we've talked about with another groundbreaking technology, and it's the computer industry. So the blue curve here, again, so we have a log scale on the y-axis, and now we don't have centuries, we have decades down here. So something happened, I think around 1960, and that was the invention of the microchip. And ever since then, our ability to put transistors on microchips has doubled in a little less than 2 years. That is what most of you probably know as Moore's law. So we've had an exponential growth in the power of computing over the past 50 or so years. And we all know what that has done. My students laugh at me when they say I wrote my first email when I did my master thesis. I got my first cell phone when I moved to Copenhagen when I was 18. And now everyone, I have a kid who's 8 years who has a cell phone. So this is just examples of how fast these things develop. But what's interesting in that comparison, that is that certain technologies in biology develop even faster. So we have an orange curve here that is basically available DNA information, so DNA sequence information available on the internet. So DNA sequencing was invented in the 70s, I'll get back to that in a moment. And then we've basically had exponential growth in the number of available DNA sequences in the past 30 years or so. There's actually some really interesting details in this curve. So here you get a break in this curve, which is around 1990. Not because technology developed further, mostly because there was a lot of investment in DNA sequencing. Most famous is the Human Genome Project. So around 1990, it was decided that we should sequence the human genome. And so a lot of money went into it, and the result was a lot of sequences came out. But we get another kink in the curve here, and that's because all these investments in technology led to development of new technologies to sequence. And this is what's known as next generation sequencing, which has just, really dropped the price dramatically on sequencing and increased the available sequence information on the internet. And kind of interesting that it grows faster than the computer, the power of computing, right? So at some point we'll kind of run out of computer power to handle biological sequence information. So the last curve is basically our ability to write DNA or synthetically synthesize DNA, which is also moving at an exponential pace. And there's some people believing, so there's a lot of company and a lot of investment in synthesizing DNA right now. So there's sort of a strong belief that we'll get a kink in this curve, and it might actually, to some extent, outcompete the use of CRISPR-Cas9. We can just order DNA and they'll get shipped to us in an envelope. Okay, so these technologies, I kind of like this historical view on some of the most important, let's say, discoveries in molecular biology. And a really good place to start is this famous, structure of DNA solved by Watson and Crick. And it's maybe not so much the actual sort of beautiful double helix structure, which was really important in this discovery, but there's really probably the most, let's say, understated sentence in any paper in science literature. It does not escape our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So they really found out how genetic information can be transferred from one generation to another in DNA. So this is sort of the alphabet invented for biology in around 1950, and so 10 years later, Watson and Crick got the Nobel Prize for this work. So we get these decades jumping in technology development. So it took roughly 10-15 years after we actually knew that genetic information was coded like that in DNA and transferred like that, to actually be able to get the language, the genetic code solved. And this was work done mainly by a guy called Marshall Nirenberg, who actually found out that it takes 3 nucleotides in DNA to encode an amino acid in a protein, so the genetic code. So Nirenberg shared the Nobel Prize with 2 other chaps, including a guy called Hargo bin Khorana. And so he developed some technologies that were really important for elucidating the genetic code. In fact, what Har Khorana did was that he was the first to synthetically synthesize DNA, which is it's kind of an incredible thing. So this is kind of the chemistry of the molecules of life, and now you can take simple chemicals and do chemistry, and you can create the language of living things. And he was the first one to do that. What about reading? So we've heard about Sanger sequencing, which is called like that because Fred Sanger invented it in the early 70s and got his second Nobel Prize, by the way, for this technology, which is pretty cool. Something else really important happened in the 70s. So we discovered restriction enzymes. And so restriction enzyme is kind of DNA editing 1.0. So that enabled us for the first time to cut and paste DNA, DNA molecules. And interestingly, restriction enzymes is also a defense system from bacteria that we, beautifully heard about in the first intro that CRISPR-Cas9 is as well. But restriction enzymes is a little bit more crude, not as elegant as CRISPR-Cas9. So instead of being able to sort of copy any DNA sequence and become immune to any phage attack or whatever, restriction enzymes basically always recognizes the same nucleotide sequence. And so when it sees that sequence coming from outside, it will cut it, and whatever is invading that DNA will die and not invade the microorganism. This is, of course, basically you could say a good starting point for biotechnology. And probably the most famous example of that is the first cloning using restriction enzymes of human insulin and putting it into bacteria and producing recombinant insulin to treat diabetes. And so this was done around 1980 using restriction enzymes as a technology. And of course that has changed the life of a lot of diabetic people. it has changed the life of us in here and people in Denmark, because Novo Nordisk is making a lot of money, and it gave me a job because I'm working at the Novo Nordisk Center for Biosustainability. These chaps, of course, also got the Nobel Prize. So I already talked about synthetic DNA. So this was done primitively in the 60s, but was really refined in the late 70s by something called solid-phase phosphoramidite chemistry. This really enabled synthesis of small pieces of DNA at a very cheap and very efficient way. So now, these days, you can buy DNA, short pieces of DNA, these so-called primers or oligonucleotides, at a really low price. And in combination with an enzymatic reaction, Kary Mullis actually used these small oligonucleotides to invent the so-called polychain reaction. So the polychain reaction allows you to take small pieces of DNA and have a long template of some, at a very low amount, and then exponentially amplify that. So this really enables you in the lab as a molecular biologist to work with DNA in a practical fashion using test tubes. It also basically defined the field of forensic genetics. For those of you who see CSI, that's basically based on this technology, right? So the ability to go to a crime scene, pick out a little piece of blood, amplify it, millions and millions of time, and then read it, and then see who was the murderer. So Kary Mullis, only a few years after inventing PCR, it was so clear that that was such a disrupting technology for the molecular biologist that he himself received the Nobel Prize in 1993. Okay, so fast forward to CRISPR-Cas9. We've heard a lot about what CRISPR-Cas9 is doing, so I'm not going to give you the details of that. I believe it belongs in the same category as these other inventions. I bluntly put 3 question marks here. There's probably no question that 3 people will receive the Nobel Prize for this. I bluntly also put 2001 here, something. So if anyone wants to take a bet, 16, 17, 18, and 19, we will get a Nobel Prize for CRISPR-Cas9. Anyone? I already lost it twice because I thought they would get it 2 years ago and this year. But now I'm safe betting on 3 years. So the reason we are here, if you go into Google Trends and put in CRISPR, you get a graph like this. So you get an exponential amplification of interest in CRISPR. You can go into PubMed and look at the publications. And again, you have sort of an exponential increase in the number of publications that uses CRISPR. So this is really showing, just plain statistics, how important this technology is becoming. Okay, so at the last part of my talk, I used bluntly this titled Reverse Genetics 2.0. So what do I mean by that? So genetics is basically the studies of the sort of relationship between a certain function, a certain phenotype of a living system, and the genes that are responsible for that. So classical forward genetics basically is you have a certain phenotype. I have brown hair. What genes are actually responsible for me having brown hair? So in reverse genetics, you're more interested in the genes from the start. So basically, what is this gene doing? And a classical way to find out what gene is doing is to delete it, that we also heard a lot about today. So, making a frame shift or removing a gene completely, and then look for phenotypes of your living system. And this way you learn a lot about basic science about what certain genes do in living organisms. So, in the few examples I'm going to show you from our lab is sort of reverse genetics examples where we are, trying to understand what specific genes are doing by disrupting their expression or removing them from the living cell. So, at the center, we work with many different organisms. We work with CHO cells, mammalian cells. We work with yeast. I'm in the bacterial section. And since there hasn't been any sort of one today talking about bacteria, I want to give you 2 examples. So there's something special about bacteria in terms of CRISPR-Cas9. And that is that for once we were after the other living cell system in terms of developing technologies. The reason I really love working with E. coli as a model organism is it's so fast, it's so easy, you can sort of explore an incredible amount of really bad ideas that I usually get, and you can kill them the next day because that's how fast it is to do an experiment in E. coli. If you work with eukaryotes, it takes years, so I'm not patient enough to do that. But it took a little longer to actually find out how to use CRISPR-Cas9 in bacteria. And that's basically because a bacteria like E. coli doesn't have non-homologous end joining or homologous-directed repair. So basically, if you target CRISPR to a certain sequence in a bacteria, all you do is you kill it. So that's not really very useful. So for that reason, CRISPR-Cas9 has been repurposed a bit compared to what we've heard about today for use in bacteria. So basically, it has been repurposed for either selection or for interference instead of knocking out genes. So selection meaning that we use another technology to do a change in DNA. And this is usually very inefficient, at least if you do it on the genome in bacteria. So we need to have a selection method to find those that actually were changed. So basically, then we use CRISPR to kill the wild-type DNA sequence, and then the only survivors is this rare event where we actually change the DNA sequence. We can also use it for interference, so basically you can repurpose CRISPR-Cas9 in all living organisms to use a catalytically dead Cas9, so it no longer cleaves DNA, it will just bind specifically to the DNA sequence that you choose based on your guide RNA design. And so what happens when you do that, I don't think it's really probably known, but you can sort of imagine that if you have transcription and you have your RNA polymerase wanting to do a messenger RNA and you have your Cas9 sitting there, it's no longer able to express the gene, right? So that's probably what happens when you use a catalytically dead Cas9 targeted to a specific gene. So you don't knock out the gene, it's still there, but you knock down the transcription and the expression of the gene, which is really useful in many ways. So example of that from the lab, and this is work done by Ida, who's in the audience, and I promise not to point her out. Where are you, Ida? Okay, but if she stands up, then you can go talk to her after. There she is. So basically, Ida has been studying essential genes in E. coli, and the problem with essential genes is that you can't do reverse genetics, right? Because if you knock it out, you kill the cell. So what can you do? We came up with two solutions to that, and that is we use CRISPR-I, so we sort of dampen the expression of the gene, but that leaves you with one problem, and that is the protein is still there, right? It was synthesized, and that's why E. coli survived. So at the same time we're actually destabilizing the protein. So at a certain time, we grow up E. coli and now say, okay, now we want to learn what this gene and protein is doing. So then we stop transcription, we remove the protein, and then we can study E. coli. So we did this is a very simple example. This is just fluorescent protein, and basically we grow it up, and we get more and more fluorescence in our cells, and then we induce the guide RNA to stop transcription along with the dead Cas9. And then we have a protease that will actually lead to degradation of the protein at the same time. So we induce those things, and you can see when we don't induce anything, we get increase in fluorescence. When we induce it with two promoter inducers here, we get a complete loss of fluorescence. This was just a proof of concept that the technology worked. So what can we use this for? So in this case, what we've done is that we've targeted an essential gene. So no one has ever been able to knock out this gene before. But here we can see what happens when so these are actually two different essential genes in E. coli. So when you just grow E. coli up and plate them at a certain dilution, you get a carpet of bacteria on your plates. But then when you actually induce the system that goes in and interferes with this gene and degrades the protein, you can see when we only induce CRISPR, we get much fewer colonies, so very few survivors by treating it with the CRISPR system. When we do both the CRISPR and degrades the protein, then we get basically no surviving, only very, very few surviving colonies. So we've been able to sort of almost completely remove the activity of this protein, which can be really important for studying what it's doing in the cell. It can also have an applied perspective. So the development of new antibiotics these days is really important. It's a major problem in the world that we are running out of antibiotics. So a common way to discover new antibiotics is you throw hundreds of thousands of compounds on a bacteria, and then you see what compound will kill it. The problem is when you throw 100,000 compounds, you cannot control. You cannot do that at a very high concentration. So you may miss some hits in that screen where it's actually inhibiting the cell from growing, but you're using too little of the compound. So what you can do is you can make hypersensitive bacteria. You can lower the levels of the essential genes or proteins, and that makes them more sensitive to antibiotics. And now you get more hits when you throw your library of antimicrobials on your cells. So this is what we've been showing in this graph. Basically, when we are targeting this gene MUR-E, we get much more growth inhibition with using this antibiotic called fosfomycin. So only 2 or 3 slides between you and dinner or snacks. So this is an example performed in the Naval Lab by in Alex Nielsen's lab, and performed by Song, a recent graduated PhD student. And what they actually did was really cool. They've actually been able to create a library with guide RNAs against all the genes in E. coli. So you can actually imagine, you can do this pretty cheaply by just ordering the oligonucleotides that corresponds to creating the guide RNAs and that targets all these genes. And now you've created a potential library that can knock out all genes in E. coli. So you throw that at E. coli, and then you look for certain phenotypes that might be attractive. This could be higher production of a chemical that we typically do at the center, higher production of a protein, I don't know, green color or faster growth, you name it. Any phenotype you can think of, now we have, in a very cheap fashion, been able to more or less knock out or knock down all genes in E. coli. So the concluding remark is, CRISPR-Cas9 is groundbreaking technology. It's a fantastic tool. We've heard a lot about the tools that this can be used for today. It's actually, interestingly, also even changing our sort of general view on biology. So probably, let's say, the most central paradigm in biology is evolution. I hope you will agree with that. And when we think about evolution, most of us think about Charles Darwin and natural selection, survival of the fittest. But the concept of evolution was first actually introduced by Lamarck around, 50 to 100 years before Darwin. So he was the first who sort of came up with the concept that living organisms evolve. The reason that his theories didn't really catch on was that people always think that he's kind of ridiculous in the way he thought evolution would work. The most famous example is this Rudyard Kipling childhood book with the giraffe and how it got its long neck. That's basically based on Lamarck theory. So basically it got this long neck because it's reaching out for the high branches in the tree, and so it gets a long neck and it transfers that trait to its next generation. So the environment will tell the genome in modern language to grow a longer neck. Okay? But this is, of course, absolutely wrong, and then Darwin came up with a much better solution to this, and that is that you probably have random changes on the genome, and then, once in a while you get a mutant giraffe that has a longer neck, and over a long, long, long time, those will have some benefits compared to the short necked giraffe, so they will take over the population. Okay, so most people in this field believe mostly in Darwinian evolution and not in Lamarckian evolution. If you look at the, sort of the gene or the DNA level version of this, Darwin would say, well, you have random mutations to a gene, but then you have environmental factors that select for those that are beneficial. Okay, many different mistakes, but only one survives, and that's the one that gives you a positive trait. Whereas Lamarck says, no, sometimes you actually have specific environmental factors that gives you a specific change, and then it's simply transferred to the next generation. So the last remark I want to say today, it's actually interesting that CRISPR-Cas9 has revived Lamarck a bit, because this is exactly what CRISPR-9 did in bacteria, right? So, bacteria is infected by a phage or a plasmid, and then it cuts out a piece of the DNA, puts it on the genome. This is transferred to the next generation, and the next generation is now more resistant to a phage attack or plasmid attack. So that's Lamarckian evolution. So we have to, and there are many other examples than CRISPR-Cas9, but it's a really nice example that some of Lamarck's ideas about evolution was actually not that crazy. So thanks for your attention. I always have to acknowledge funding from Novo Nordisk and my fantastic research group. Questions, I'm happy to take them, or email me at this address.